advanced systemic mastocytosis is a group of rare diseases, but we really don't have a very good treatment for them. So let's talk about some alternatives. We're with Dr. Daniel J. DeAngelo, who is an MD and a PhD. He is the Director of Clinical and Translational Research at the Adult Leukemia Program at the Dana-Farber Cancer Institute. Now, the only currently approved agent to treat advanced systemic mastocytosis is midostaurin, right?
That's correct.
And that's not ideal.
Well, midostaurin is a multi-kinase inhibitor. It hits a lot of targets.
Yes.
Yeah, and it does have some toxicity associated with it, but it works. It has a complete and partial remission rate of about 17%, and so it's better than nothing.
Yes, true.
And it does improve the quality of life in some patients who can tolerate it. There's some GI toxicity associated with some nausea and vomiting, but it is an agent that has some applicability, but it's nice to have better alternative strategies.
So tell me about BLU-285.
Sure. BLU-285 is a more potent KIT inhibitor, number one. Number two, it's targeting the mutant form of KIT, which is the D816V, which is the driver of the disease process in about 90% of patients with systemic mastocytosis. So it's actually hitting the target a little bit more potently and a little bit more effectively.
So for the phase 1 data you're presenting here at ASH, describe the eligible patients and then how they were treated.
Sure. So it was a phase 1 study, so we try to include a lot of patients in the study. We enrolled 32 patients with WHO diagnostic criteria for systemic mastocytosis, as well as other refractory myeloid malignancies. Vast majority of the patients had systemic mastocytosis. And all 32 patients actually had some response, which was really exciting in a phase 1 clinical trial. We never established a maximum tolerated dose. We established 300 milligrams once a day as the best acceptable dose, and we're taking that through to a phase 2 study.
Now, there were a few different KIT mutations represented in your study, patients, including 3 with no detectable KIT mutation?
No detectable KIT D816V mutation, that's correct, because we included other patients in the phase 1 portion or the dose escalation portion in order to rapidly accrue patients. But the vast majority of patients had a KIT mutation, and those are the patients that were taken through in the expansion cohorts as well in subsequent studies.
So with no KIT alteration, what made you think that these 3 might respond?
Oh, I mean, it was a phase 1 study, so these are patients that had relapsed refractory myeloid malignancies or had systemic mastocytosis. The KIT is not, the KIT driver is 90%. The D816V is 90% of patients with systemic mastocytosis. It's not the only game in town.
Right.
And the BLU-285 will also hit other mutations as well as some wild type. It's just more effective against the driver.
Also, most of the patients had received prior anti-neoplastic therapy, so this was a tough crowd.
Yeah, 70% had had prior therapy, including 4 patients who had progressed on midostaurin.
Okay, because this was a phase 1 study, your primary objective was to define the dosing and safety. So what did you learn? You've got the dosing. What did you learn about safety?
Well, we didn't see a lot of toxicity. So 30 of 32 patients remain on study, which is kind of unusual in a phase 1 study. No patient came off study for toxicity concerns. Most of the toxicities were lower grade, which we consider grade 1 and 2. And the grade 3 toxicities were usually edema or fatigue, which were easily controlled with either dose delays or reductions. So it was extremely well tolerated, and there were 0 patients, as I mentioned, who came off for toxicity concerns.
How fast is the absorption?
It's rapid, so it's a once-a-day, single-day dosing, and so it's very amenable to outpatient and continuous dosing. So it really wasn't an issue.
So what preliminary anti-neoplastic activity did you see? Were you looking at any of that?
We were, as a secondary endpoint, and to our excitement, we saw basically a large percentage of patients who responded. So all 32 patients had reductions of more than 50% of their serum tryptase level, which is a good marker of overall mast cell burden. 25 patients had bona fide marrow disease that we could follow. And 60% of those actually cleared their marrow entirely, had a complete remission with respect to their marrow disease. So looking at a subset of patients that were evaluable by the IWG criteria, we were able to show that the response rate was 72%, with 56% of patients having either a complete or partial remission. So very high for a phase 1 dose escalation study, if you will.
So what's next?
So the next is to confirm the side effects and to confirm the efficacy by using, looking at dose expansion cohorts with specific disease criteria. looking at 15 patients in aggressive systemic mastocytosis with associated hematologic neoplasia, a related disorder, and mast cell leukemia, 5 patients, just to confirm what we saw in the dose escalation port. Then there are planned phase 2 confirmatory trials next year, both in aggressive systemic mastocytosis, but also a dose-finding study for patients with less aggressive disease called indolent or smoldering systemic mastocytosis. So those are planned for next year.
So my guess is we'll be talking to you again down the line, not too probably long from now.
I hope so.
Terrific. Thank you very much.
Thank you.
And for ASH 2017, please look around. We have a lot of coverage in ASH Clinical News as well as online. I'm Rick McGuire.